Hello dear students. We'll continue from the last video wherein I was explaining about the histones and how exactly the histones are going to be arranged into an octameric core around which the DNA of 145 base pairs are going to be wrapped around with 1.5 turn 1.7 turn, and then uh, the presence of H1, which is going to be presented the linker DNA, which is connecting the two nucleosomal structures and stabilizing the linker part of the DNA molecule and all those things. And even I just gave you an insight like how the transcriptional repression is going to be happening because of the presence of nucleosome, and how this transcriptional repression is going to affect the transcriptional ability in case of eukaryotes. So where you can see a nucleosome free, if you remember, a nucleosome free DNA, whatever the relative transcriptional rate will be there, it will be reduced by almost 75% when in case of a nucleosomal DNA molecule, because the promoter regions are the gene portion. Sometimes the promoter regions are going to be covered within a nucleosome, or even sometimes we can find the promoter regions may be free in the linker DNA, but the gene portions will be covered by the nucleosomes. In both the case, to have an efficient transcriptional process, so we should see that this particular DNA is going to be free, then only an efficient transcriptional process can take place. So in case of this concept of nucleosomal positioning, it is nothing but the nucleosome structures, so their position is going to act means plays an important role as per earlier discussion in the process of transcription. And this nucleosome positioning can act as an anti-repression activity. So wherein whenever the transcriptional activation is required, the nucleosome anti-repression is going to take place by, it can happen either by removing the nucleosomes which are going to be obscuring the promoter regions, means covering the promoter regions, or even it can also happen by preventing the initial nucleosomal binding to the promoters, means the promoters either they are going to be get set free from the nucleosomes, or it can also happen that the nucleosome is going to be going away from that particular promoter region, so which is going to lead to repositioning of the nucleosome and acts as an anti-repressive activity. So both the actions, that is again and generally this particular nucleosome is going to position itself back once the transcription is going to be completed. So both the actions are forms of nucleosomal positioning. So this particular nucleosomal position is going to happen due to the binding of activators. They force the nucleosome to take up a position around or around the promoter, so that the promoter will be available for the transcription factors and not within the promoter or covering the promoter. So this particular experimentations was done in order to confirm this, the nucleosome positioning. It will lead to the formation of nucleosome free zones, particularly at the control regions of the genes, and this particular action was means this particular component or point concept was well understood when people did an experiment by using SV40 DNA. What they have done is they have taken SV40 mini chromosome, and they performed an experiment to check the existence of nucleosome free zones, and they did it by using restriction enzymes. They took the sp40 mini chromosome and subjected for restriction enzyme like BGL1, BAMH1, ECOR1, and these restricted fragments were checked, and it was established that there will be nucleosome free zones are going to be seen whenever there is going to be gene transcription is going to take place. Then another experiment the people have done in order to identify, which also confirmed this, by wherein they have checked for DNA's hypersensitive regions, that is the regions of DNA which are going to be sensitive for the activity of DNases. As we know, DNases can able to bind, react, means cleave the DNA only when the DNA is available freely, but if it is going to be covered with the nucleosomes, then that particular DNA will not be able to get cleaved by the DNases. So you can see what they have done is they have taken the DNA, they have taken the a particular micro mini chromosome having in one case you can say promoter region is covered by the nucleosome, on the other case also is present in between. They have taken this 13 base pair, kilobase pairs fragment of DNA and subjected for restriction enzymes. When they subjected for restriction enzymes, what they found is, so this restriction enzyme has cleaved the DNA molecule, the nucleosomal DNA has got cleaved in one case, the other case it did not got cleaved, because the cleavage was possible in one case because there is a small amount of DNA which was free, where the DNAs is actually bound and cleaved. So this was confirmed when they subjected this particular DNA for removal of protein molecules by using salt concentration, and that DNA was run in the agrosial electrophoresis, showing in one wherein in one particular they have even amplified with a probe and then they subject for separation. So in one case wherein the gene DNA was inactive, not involved in the process of transcription, they found only one band because restriction enzyme or the DNases could not able to cleave, whereas in the other case they found formation of two fragments of 7 and 6 base pairs. That indicates that this particular the mini chromosomal DNA with the nucleosomes was involved in the process of transcription. Because of that, there was creation of a DNA hypersensitive region, so which got acted upon by the restriction enzymes and cleaved the particular fragment into a 6 kilobase pairs and 7 kilobase pairs fragment, and it was missing of 13 kilobase pairs fragment. So that confirmed that these regions which are during a particular process of transcription, some of the part of the DNA is going to be set free, so that the transcriptional factors can able to bind, and as well the transcription of the gene can easily takes place. So now how this particular whole of the things are means all these particular processes are going to take place. This is going to start with the modification of histones, and this modification of histone is going to act as a code for which particular portion of the DNA need to be undergoing the modifications, or in other words chromosome modification is going to be required in order to make the DNA available for the transcription, and as well as in order to see that the DNA which was set free for transcription, now even it can be covered once the transcription is going to be over. So this histone modification starts with the process of histone acetylation. So James Brownwell and David Alice, they succeeded in identifying and purifying the histone acetyl transferase enzyme. This is the enzyme which transfers the acetyl group from a donor that to a particular core histones. So core histones are nothing but H1, H2A, H2B, okay, dimer two of the dimeric structures, and then the H3H4 tetramer. So all together they are going to form an octameric core structure around which the DNA is going to be wrapped. So these investigators, they used a creative strategy in order to isolate this particular HAT enzyme. What they have done is they started with tetrahymena cells, because this particular organism has histones that are heavily acetylated, okay, and that means high concentration of HAT should be available in that. So they prepared the extracts from the micronuclei and from the macronuclei and subject them for gel electrophoresis, and then in an SDS gel impregnated with the histones. In order to detect the hat activity, what they have done is they soak the gel in the solutions containing acetyl CoA, which is radioactive labeled acetyl group, and after that they may, means if the hats are active they are going to acetylate the histones, and if it is inactive they cannot able to acetylate the histones. And these particular gel, when they have actually checked for autoradioactivity and all these things, they found that the acetylation of the histones happened, and this acetylation was carried out by a band of hat activity corresponds to P55, a protein 55 kd in size was seen in the gel. So accordingly these people named as P55, that is protein 55 or polypeptide 55. The amino acid sequence is preferred is in sorry amino acid sequence inferred from the base sequence of P55. So it is very similar amino acid sequence as of yeast protein called GNC5 polypeptide, and they call them as GNC5 polypeptide family. And this GNC5 has been identified as an coactivator in acid transcriptional activator such as GNC4P. So the amino acid sequence similarity suggested that both P55 and GNC5 polypeptide protein and are had protein molecules, they involve in the gene activation. And in order to verify that GNC5 P and has a hat activity, even Ellis and colleagues, what they did, they expressed this particular gene unequally and then subjected and P55 to an SDS page activity gel assay which confirmed this particular activity. So this is the acetylating how it is going to happen. You can see the lysine is going to get acetylated by the acetyl transferase enzyme into epsilon N acetyl lysine. So acetylation generally happens to the lysine amino acids or even the other amino acids like so N-terminal tails of these amino acids, they are going to get reversibly acetylated, okay, particularly of H3 and H4, and the consequences of acetylation is that acetylation, which is going to change the amino acid of the lysine and leads to reduce the positive charge, and this reduction in the number of positive charge, that going means confirms sorry this particular positive charge is going to get decreased, and due to this decrease in the positive charge, the interaction between the DNA and the histone is also going to get reduced, and it leads to a change in the conformation of nucleosome and destabilizes the internucleosomal contacts. So this leads to the removal of histones tail from the DNA and improves the availability of DNA for the binding of transcription factors. So that is how this modification acetylation is going to plays an important role. You can see here histone is going to get acetylated so people have found this histone acetylation is going to have a very important link in the process of transcription. What they have found is the acetylated histones tails enriched in transcribed chromatin. So by the process of immunofractionated chromatin, so acetylated fractions enriched in an active chromatin, that is transcriptionally very much active chromatin, they found acetylated histones are very high, and the histones are going to get neutralized, mutation is going to be there at the tail, they found the mutations increases, which leads to less acetylation, is going to affect the transcription. So this was confirmed. People used a method called chromatin immunoprecipitation method. Okay, so that is the chip it's called as. It shows that in an active promoter there will be more number of acetylated histones, and acetylation makes the nucleosomal DNA more accessible for transcription factor binding, and also has a role in potentiation of that process. So I'll stop here, continue other aspects or this particular hat and other modifying enzymes and modifications in the coming class. You can see these are all acetylated histones and the positions. So thank you.